Microbial diseases of the digestive system. Contents. Review the structure and function of the system. Normal microbiota present. Bacterial diseases, viral diseases, fungal diseases, parasitic diseases. Microbial diseases of the digestive system are the second only to respiratory diseases as causes of illness in the United States. Most such diseases result from ingesting food or water contaminated with pathogenic microorganisms or their toxins. These pathogens usually enter the food or water supply after being shed in the faces of people or animals infected with them. Therefore, microbial diseases of the digestive system are typically transmitted by fecal-oral cycle. This cycle is interrupted by effective sanitation practices in food production and handling. Modern methods of sewage treatment and disinfection of water are essential. There is also an increasing awareness of the need for new tests that will rapidly and reliably detect pathogen in food. The purpose of digestive system is to digest food, that is to break them down into small molecules that can be taken up and used by body cells. In a process called absorption, these end products of digestion pass from the small intestine into the blood or lymph for distribution to the body cells. Then the food moves through the large intestine where water, vitamins, and nutrients are absorbed from it. Over the course of an average lifespan, about 25 tons of food pass through the GI tract. The resulting undigested solid called feces are eliminated from the body through the anus. Intestinal gas or flatus is a mixture of nitrogen from swallowed air and microbially produced carbon dioxide, hydrogen, and methane. On average, we produce 0.5 to 2.0 liters of flatus every day. Normal microbiota present. Bacteria heavily populate most of the digestive system in the mouth. Each millimeter of saliva can contain millions of bacteria. The stomach and small intestine have relatively few microorganisms because of the hydrochloric acid produced by the stomach and the rapid movement of food through the small intestine. By contrast, the large intestine has enormous microbial population exceeding 100 billion bacteria per gram of feces. Up to 40% of fecal mass is microbial cell material. The population of the large intestine is composed mostly of anaerobes and facultative anaerobes. Most of these bacteria assist in the enzymic breakdown of foods, especially many polysaccharides that would otherwise be ingestible. Some of them synthesize useful vitamins. The small intestine also contains important antimicrobial defenses. Most significantly, millions of specialized granule-filled cells called Paneth cells. These are capable of phagocytizing bacteria, and they also produce antibacterial proteins called defenses and antibacterial.
I'm going to report the different bacterial diseases of mouth and digestive system. Bacterial diseases of the mouth. Dental caries or tooth decay. The teeth, unlike any other exterior surface of the body, are hard and do not shed surface cells. These allow masses of microorganisms and their products to accumulate. These accumulations, called dental plaque, or a type of biofilm and are intimately involved in the formation of dental caries or tooth decay. Oral bacteria convert sucrose and other carbohydrates into lactic acid, which in turn attacks the tooth enamel. The microbial population on and around the teeth is very complex. Probably the most important cariogenic, the dental caries causing bacterium is Streptococcus mutans, a gram-positive coccus that has important virulence characteristics. Even people who avoid tooth decay might, in later years, lose their teeth to periodontal disease, a term for a number of conditions characterized by inflammation and degeneration of structures that support the teeth. The roots of the tooth are protected by a covering of specialized connective tissue called cementum. As the gums recede with age or with overly aggressive brushing, the formation of caries on the cementum becomes more common. One of the diseases of periodontal is gingivitis. In many cases of periodontal disease, the infection is restricted to the gums or gingivae. This resulting inflammation, called gingivitis, is characterized by bleeding of the gums while the teeth are being brushed. This is a condition experienced by at least half of the adult population. It has been shown experimentally that gingivitis will appear in a few weeks of brushing if brushing is discontinued and plaque is allowed to accumulate. An assortment of streptococci, actinomycetes, and anaerobic gram-negative bacteria predominate in these infections. Periodontitis is an insidious condition that generally causes little discomfort. The gums are inflamed and bleed easily. Sometimes pus forms in periodontal pockets surrounding the teeth. As the infection continues, it progresses toward the root tips. The bone and tissue that support the teeth are destroyed, eventually leading to loosening and loss of the teeth. Periodontitis can be treated surgically by eliminating the periodontal pockets. Acute necrotizing ulcerative gingivitis or Vincent's disease or trench mouth. It is one of the more common serious mouth infections. It causes enough pain to make normal chewing difficult. Foul breath or halitosis also accompanies the infection. Among the bacteria usually associated with this condition is Prevotella intermedia, averaging up to 24% of the isolates. Because these pathogens are usually anaerobic, treatment with oxidizing agents, debridement, and the administration of metronidazole or antibodies may be temporarily effective. The bacterial infections of the gastrointestinal tract. Enterovirulent Escherichia coli. E. coli is a gram-negative bacillus that is found in the GI tract of all humans. The strains and serotypes of E. coli that are part of the indigenous microflora of the GI tract are opportunistic pathogens. They usually cause no harm while in the GI tract but have the potential to cause serious infections if they gain access to the bloodstream, the urinary bladder, or a wound. E. coli is the major cause of septicemia, UTIs, and healthcare-associated infections. Bacterial foodborne intoxications or foodborne infections, food poisoning. The term food poisoning is broad and may include diseases resulting from the ingestion of chemical contaminants as well as bacteria or bacterial toxins, picotoxins, mycotoxins, viruses, or protozoa. Technically, diseases resulting from the ingestion of toxin-producing microorganisms are called infectious diseases, whereas disease resulting from the ingestion of preformed microbial toxins are called microbial intoxications. The distinction is based on where the toxin is actually produced in the body in vivo or in the food in vitro. The incubation time, the time that elapses between ingestion and onset of the symptoms, is usually shorter in microbial intoxications. If toxin producing bacteria are ingested, the incubation time will will depend on the number of bacteria ingested, their generation time, and the amount of time it takes them to produce enough toxin to produce symptoms. Enterovirulent Escherichia coli. These are There are other strains and serotypes of E. coli in nature that are not indigenous microflora of the human colon and always cause disease when they are ingested. Collectively, these strains and serotypes are referred to as enterovirulent E. coli. Information pertaining to two genotypes, the enterohemorrhagic E. coli and the enterotoxigenic E. coli. Enterohemorrhagic E. coli or EHEC diarrhea. This disease consists of a hemorrhagic watery diarrhea with abdominal cramping. Usually, patients have no fever or only a slight fever. About 5% of infected people, especially child younger than age 5 and the elderly, develop hemolytic uremic syndrome with anemia, low platelet count, and kidney failure. Patient care. Use standard precautions of hospitalized patients. Add contact precautions for diapered or incontinent patients. Pathogens: E. coli 0157H7, a serotype that possesses a cell wall antigen designated 0157 and a flagellar antigen designated H7, is the most commonly involved EHEC serotype. Reservoirs and mode of transmission. Reservoirs include cattle and infected humans. Transmission occurs via the fecal-oral route, inadequately cooked, fecally contaminated beef, unpasteurized milk, person-to-person contact, or fecally contaminated water. Laboratory diagnosis: E. coli 0157H7 infection should be suspected in any patient with bloody diarrhea. Stool specimens should be inoculated into sorbitol McConkey agar. Colorless sorbitol negative colonies should then be assayed for 0157 antigen using commercially available anti-serum. Enterotoxigenic E. coli or ETEC diarrhea, traveler's diarrhea. This disease consists of a watery diarrhea with or without mucus or blood, vomiting and abdominal cramping, dehydration, and low-grade fever may occur. Enterotoxigenic strains of E. coli are the most common cause of traveler's diarrhea worldwide and a common cause of diarrheal disease in children in developing countries. Once contracted, the best treatment is the usual oral rehydration recommended for all diarrhea. In severe cases, antimicrobial drugs may be necessary. Prescribed antibiotics may provide some protection. Another option is to take bismuth-containing preparations such as Pepto-Bismol, but the best advice in risky areas is to prevent infection. Patient care. Use standard precautions for hospitalized patients. Add contact precautions for diapered or incontinent patients. Pathogens. ETEC diarrhea is caused by many different serotypes of enterotoxigenic E. coli that produce a labile toxin, a heat stable toxin, or both. Reservoirs and mode of transmission. Infected humans serve as reservoir. Transmission occurs via the fecal oral route, ingestion of fecally contaminated food or water. Laboratory diagnosis. ETEC diarrhea is diagnosed 5101 isolation of the organism from stool specimens, led by demonstration of enterotoxin production, molecular diagnostic procedures, or immunodiagnostic procedures. Bacterial gastritis and gastric ulcers. Infection with Helicobacter pylori can cause chronic bacterial gastritis and duodenal ulcers. Gastritis is suspected when a person has upper abdominal pain with nausea or heartburn. People with duodenal ulcers may experience gnawing, burning, aching, mild to moderate pain just below the breastbone, and an empty feeling and hunger. The pain usually occurs when the stomach is empty. Drinking milk, eating, or taking antacids generally relieves the pain, but it usually returns 2 to 3 hours later. Gastric ulcers and gastric adenocarcinoma are also epidemiologically associated with pylori infection. Gastric ulcers and duodenal ulcers are type of peptic ulcers. Complications of peptic ulcers include penetration, perforation, bleeding, and obstruction. Patient care. Use standard precaution for hospitalized patients. Pathogen. H. pylori is a curved microaerophilic capnophilic gram-negative bacillus that is found in the mucus-secreting epithelial cells of the stomach. No other bacteria are known to grow in extremely acidic stomach. Reservoirs and mode of transmission. Infected humans serve as reservoirs. Transmission probably occurs via ingestion, presumed to be either oral to oral or fecal to oral transmission. Laboratory diagnosis: the urea breath test, NH excretion test, molecular diagnostic processes, and immunodiagnostic procedures are among the diagnostic techniques. The patient consumes radioactive urea and then has his or her breath evaluated 60 minutes later to radioactive CO2 because H. pylori produces the enzyme urease, which breaks down urea into ammonia and CO2. Radioactive CO2 shows the presence of H. pylori in the NH test. The patient swallows urea laced with radioactive nitrogen. The ammonia produced by H. pylori in the stomach is taken into the circulation and released in the urine. Campylobacter enteritis. Campylobacter enteritis is an acute bacterial disease ranging from asymptomatic to severe with diarrhea, nausea, vomiting, fever, malaise, and abdominal pain. The disease is usually self-limiting, lasting to 2 to 5 days. Stools may contain gross or occulted blood, mucus, and WBCs. Patient care: use standard precautions for hospitalized patients and add contact precautions for diabetic and incontinent patients. Pathogens: the etiologic agents of Campylobacter enteritis are Campylobacter jejuni and less commonly Campylobacter coli. Campylobacter spp are curved, S-shaped, and spiral-shaped gram-negative bacilli often having a gull-wing morphology, a pair of curved bacilli following cell division. They are microaerophilic and capnophilic with an optimal growth temperature of 42 degrees Celsius. Reservoirs and mode of transmission. Reservoirs and are animals including poultry, cattle, sheep, swine, rodents, birds, kittens, puppies, and other pets. Most raw poultry is contaminated with C. jejuni, thus necessitating proper methods of cleaning and disinfecting in the kitchen. Transmission occurs via ingestion or of contaminated food, examples chicken and pork, raw milk or water, contact with infected pets or farm animals, or contaminated cutting boards. Laboratory diagnosis. Diagnosis depend on the recovery of Campylobacter spp from stool specimens using selective medium Campi blood alcohol, which contains several antimicrobial agents to suppress growth of other bacteria. Cholera. Cholera is an acute bacterial diarrheal disease with profuse watery stools, occasional vomiting, and rapid dehydration. If untreated, circulatory collapse, renal failure, and death may occur. Patient care. Use standard precautions for hospitalized patients, and add contact precautions for diapered and incontinent patients. Pathogens. The etiologic agents of cholera are certain biotypes of Vibrio cholerae serogroup 01. These are curved, comma-shaped, gram-negative bacilli that secrete an enterotoxin, a toxin that adversely affects cells in the intestinal tract called choleragen. Reservoirs and mode of transmission. Reservoirs include infected humans and aquatic reservoirs, copepods, and other zooplankton. Transmission occurs via the fecal to oral route, contact with feces or vomitus or of infected people, ingestion of fecally contaminated water or foods, especially raw or undercooked shell, fish, and other seafood, or mechanical transmission by flies. Laboratory diagnosis. Rectal swabs or stool specimens should be inoculated into thiosulfate citrate, bile sucrose, agar. Different Vibrio SPP produce different reactions of this medium. Biochemical tests are used to identify the various species. Biotyping is accompanied using commercially available antiserum. Salmonellosis. Salmonellosis is gastroenteritis with sudden onset of headache, abdominal pain, diarrhea, nausea, and sometimes vomiting. Dehydration may be severe. Salmonellosis may develop into septicemia or localized infection in any tissue of the body. Patient care. Use standard precaution for hospitalized patients. Add contact precautions for diapered and incontinent patients. Pathogens. GI salmonellosis is caused by members of the family Enterobacteriaceae, currently named Salmonella enterica, of which there are more than 2,000 serotypes or serovars. These gram-negative bacilli invade intestinal cells, release endotoxin, and produce cytotoxins and enterotoxins. Reservoirs and mode of transmission. Reservoirs include a wide range of wild and domestic animals such as poultry, swine, cattle, rodents, reptiles, example pet iguanas and turtles, pet chicks, dogs and cats, infected humans, example patients, carriers are also reservoirs. Transmission occurs via ingestion of contaminated food. Examples are eggs, unpasteurized milk, meat, poultry, raw fruits, and vegetables. Fecal to oral transmission from person to person, food handlers, or contaminated water supplies. Laboratory diagnosis. Stool specimens should be submitted to the microbiology laboratory for CNS. Salmonella spp are non-lactose fermenters and thus produce colorless colonies in McConkey agar. Biochemical tests are used for identification and commercially available antisera are used for serotyping. Typhoid fever or enteric fever. Typhoid fever is a systemic bacterial disease with fever, severe headache, malaise, and a rash on the trunk in about 25% of patients, non-productive cough, and constipation, bacteremia, pneumonia, gallbladder, liver and bone infection, endocarditis, meningitis, and other complications may occur. Patient care. Use standard precautions for hospitalized patients. Add contact precautions for diapered or incontinent patients. Pathogen. Typhoid fever is caused by Salmonella typhi, also known as the typhoid bacillus, a gram-negative bacillus that releases endotoxin and produces exotoxins. Reservoirs and mode of transmission. Infected humans serve as reservoirs for typhoid and paratyphoid, rarely domestic animals for paratyphoid. Some people become carriers following infection, shedding the pathogens in their feces or urine. Transmission occurs via the fecal to oral route, food or water contaminated by feces or urine of patients or carriers. Oyster harvested from fecally contaminated waters. fecally contaminated fruits and raw vegetables, or from processed to food by chemical transmission by flies. Laboratory diagnosis. Diagnosis of typhoid fever is made by isolation of SIV from blood, urine, feces, or bone marrow, followed by identification by biochemical test. Immunodiagnostic procedures are also available. Shigellosis or bacillary dysentery. Shigellosis is an acute bacterial infection of the lining of the small and large intestine, producing diarrhea as many as 20 bowel movements a day, with blood, mucus, and pus. Other symptoms include nausea, vomiting, cramps, and fever. Sometimes toxemia, toxins in the blood, and convulsions in children occur. Other serious complications, such as hemolytic uremic syndrome, may occur. Patient care. Use standard precautions for hospitalized patients. Add contact precautions for diaper or incontinent patients. Pathogens. The etiologic agents of shigellosis are Shigella dysentery, Shigella flexneri, Shigella boydii, and Shigella soni. They are non-motile gram-negative bacilli that are members of the Enterobacteriaceae family. And a plasmid is associated with toxin production in parallels. Relatively few 1 to 100 organisms are required to cause disease. Reservoirs and mode of transmission. Infected humans serve as reservoirs. People become infected by direct or indirect fecal to oral transmission from patients or carriers. Fecally contaminated hands and fingernails or fecally contaminated food, milk, and drinking water. Flies can mechanically transfer organisms from latrines to food. Laboratory diagnosis. Leukocytes will be present in stool specimens. Fresh, fecal, or rectal swabs should be immediately inoculated into gram-negative enrichment both and into a solid medium such as McConkey, psilocybin, deoxycholate, or hectaren enteric alcohol. Clostridium difficile or associated diseases. C. difficile is the major cause of conditions known as antibiotic-associated diarrhea and pseudomembranous colitis, which frequently occur in patients knowing antibiotic therapy. It does not seem to matter for what condition the patient was receiving antibiotics, which antibiotics the patient was receiving, the dosage, or the route of administration. Patient care. Use contact precautions for the duration of illness. Use soap and water for hand washing. The alcohol in waterless antiseptic hand wipes lacks sporicidal activity. Pathogen. C. difficile, often referred to simply as C. diff, is a spore-forming anaerobic gram-positive bacillus. Reservoirs and mode of transmission. C. difficile is a member of the indigenous microflora in about 2% to 3% of healthy, non-hospitalized adults. Hospitalized patients frequently become colonized with C. difficile as a result of its presence in the hospital environment. Laboratory diagnosis. C. difficile is identified in the laboratory using a variety of phenotypic characteristics. However, diagnosis of C. difficile associated diseases is most often accomplished using some type of commercial enzyme immunoassay or cytotoxin tissue culture assay.
Viral disease of the digestive system. Mumps. A viral infection that affects salivary glands that are located near the ears. The virus is transmitted in saliva and respiratory secretions. Ang portal of entry ng mumps is the respiratory tract. characterized by inflammation or swelling of the parotid glands, fever, and pain during swallowing. Amo niya mga symptoms sa mga tao nga naka-experience mumps. Nag-start gali ang mumps with painful swelling of one or both parotid glands 16 to 18 days after ka na-expose sa virus. Kag ginatarget ng mumps virus ang parotid glands, which is located below and in front of the ears. Parotids are also one of the 3 pairs of salivary glands of the digestive system. Hepatitis. It is the inflammation of the liver, a result of infections by other viruses such as Epstein-Barr virus or cytomegalovirus. The liver is a vital organ that processes nutrients. Gina-filter niya ang blood naton kag ginalabanan niya ang mga infection sa aton body. So, kung madamage ang liver, ang functions ni pwede maapektuhan. Heavy alcohol use, toxins, some medications, and certain medical condition can cause hepatitis. Hepatitis A vaccine-preventable liver infection caused by the hepatitis A virus or picornavirus. Found in the stool and blood of people who are infected, it is also very contagious. Symptoms: fever, headache, malaise, and jaundice. Treatment: immunoglobulin. Hepatitis A virus contains single-stranded RNA and lacks an envelope. It can be transmitted through ingestion, and if it will be transmitted, HAV multiplies in the epithelial lining of the intestinal tract. Hepatitis B, caused by the hepatitis B virus or Hepatnaviridae, a unique DNA virus, spreads when blood, semen, or other body fluids enter the body of someone who is not infected. Symptoms: similar to HAV but no headache. It will more likely progress to severe liver damage, and chronic disease will also occur. Treatment: interferon alpha and nucleoside analogs. HBV is a unique DNA virus kay instead na mag-replicate ang DNA niya directly, nagapass through pa siya sa intermediate RNA stage resembling a retrovirus. Compared to HAV, HBV is larger. Ang genome niya is double-stranded DNA and it is enveloped. Ang method of transmission is parenteral and sexual contact. As I have mentioned earlier, mag-spread ang hepatitis B if ang blood, semen, and other body fluids ng infected person maka-enter sa body ng tao na hindi infected. Acute hepatitis B, inflammation of the liver that is caused by hepatitis B virus and lasts from a few weeks up to 6 months. Symptoms: feels unwell, often suffers from low-grade fever, nausea, abdominal pain. Amo ni ang unang mga symptoms nga makita sa mga tao na may acute hepatitis B, and eventually naga-appear na ang mga symptoms such as jaundice, dark urine, and other evidence of liver damage appears. Many cases of acute hepatitis B are subclinical. In this case, some of the infected person is often unaware na may mga amoni na gali sila nga sakit. If a case of hepatitis persists for more than 6 months, the condition is considered to have become chronic. Malala na siya if ang hepatitis ng patients nagsobra na ng 6 months. Chronic hepatitis B lasts 6 months or longer. It lingers because the immune system can't fight off the infection. Most individuals suffering from acute hepatitis B clear the virus successfully. May mga patients nga nagaayo, but some feel fail to do so and develop chronic hepatitis B. Na-mention ko man kaina nga if ang acute hepatitis B maglampas 6 months, it will become chronic, so mas maging worse pagid ang sakit. Chronic hepatitis B infection may last a lifetime, possibly leading to serious illnesses such as cirrhosis and liver cancer. Hepatitis C, caused by hepatitis C virus or flaviviridae. The virus does not kill the infected cell, but it triggers an immune inflammatory response that either clears the infection or slowly destroys the liver. Since hepatitis C's method of transmission is parenteral, it is spread through contact with blood from an infected person. Hepatitis C has been described as a silent epidemic, killing more people than AIDS in the United States. Symptoms similar to HBV but more likely to become chronic. Treatment pegantiferon and ribavirin. Hepatitis D, Delta hepatitis. In 1977, a new hepatitis virus, now known as hepatitis D virus, was discovered in carriers of HBV in Italy. It can cause serious symptoms that can lead to lifelong liver damage and even death. Hepatitis D is a liver infection you can get if you have hepatitis B. It can occur as either acute or chronic hepatitis. It is also caused by hepatitis D virus or Delta viridae. Symptoms, severe liver damage. Treatment, none. Hepatitis E, spread by fecal-oral transmission or ingestion, much like hepatitis A, which it clinically resembles. The pathogen known as hepatitis E virus or Calliciviridae. It can cause the liver to swell up. Most people with hepatitis E get better within a few months, and it usually doesn't lead to long-term illness or liver damage like some other forms of hepatitis do. Symptoms similar to HAV, but pregnant women may have high mortality. Treatment, none. Viral gastroenteritis. An intestinal infection marked by watery diarrhea, abdominal cramps, nausea or vomiting, and sometimes fever. The most common way to develop viral gastroenteritis is through contact with an infected person or by ingesting contaminated food or water. Rotavirus, a genus of double-stranded RNA viruses in the family of Reoviridae. Rotaviruses are the most common cause of viral gastroenteritis, especially among infants and young children. Rotavirus cases usually peak during the cooler winter months. The first vaccine to prevent rotavirus was introduced in 1998. It was also withdrawn after serious problems developed. In 2006, a live orally administered vaccine was licensed. Treatment is usually limited to oral rehydration therapy. Norovirus, also called winter vomiting bug, a very contagious virus that causes vomiting and diarrhea. Norovirus was first identified as an outbreak of gastroenteritis in Norwalk, Ohio in 1968. The responsible agent was identified in 1972 and called the Norwalk virus. Several similar viruses were later identified, and this group were termed as Norwalk-like viruses. Fungal diseases of the digestive system. Some fungi produce toxins called mycotoxins which can affect the blood, nervous system, kidney, or liver. Ergot poisoning. Produced by Claviceps purpurea, which is a fungus causing smut infection on grain crops. A form of poisoning from ingesting rye or grains contaminated with the fungus. The toxin can restrict blood flow in the limbs, resulting in gangrene, and it may also cause hallucinogenic symptoms. Ang mycotoxins na ma-produce ng C. purpurea makakos ng ergot poisoning or ergotism, and this can happen kung makakain ka ng foods na affected ng ergot. Aflatoxin poisoning. Produced by Aspergillus flavus, which is a common mold. A condition that results from eating food contaminated with aflatoxins. The risk to human is still unknown, pero may evidence na this contributes to cirrhosis of the liver and cancer of the liver in some countries such as India and Africa. Aflatoxin can be found in many foods such as corn, peanuts, and grain, pero particular gid siya na makita sa peanuts. This can cause serious damage to livestock and can happen kung makaon nila ang mga foods nga contaminated with aflatoxin. Pag once nga natabo na, the toxins could accumulate in the livestock's system.
Parasites are living things that use other living things, like our body, for food and a place to live. You can get them from contaminated food or water, a bug bite, or sexual contact. Some parasitic diseases are easily treated and some are not. Tapeworms. Tapeworms have 3-stage life cycle. One is the egg, second, immature stage called larva, and an adult stage at which the worm can produce more eggs. Larvae can get into the muscles of their host. Infection can occur when you eat raw or undercooked meat from an infected animal. It can be eliminated using antiparasitic drugs such as praziquantel and albendazole. Types of tapeworm. One is the Taenia saginata, also known as the adult beef tapeworm. Produces only mild abdominal symptoms, and the most striking feature consists of the passage of proglottids. So proglottids is a tapeworm containing both male and female reproductive organs. Mode of transition, it is passed in the feces of an infected person, are only infectious to cattle. Humans are infected by ingestion of raw or undercooked beef, and it will develop in the intestine over 2 to 3 months. Then, Taenia solium, also known as the pork tapeworm, it produces larval stage in human host. It develops when the adult tapeworm infects the human intestine that can lead to cysticercosis. People get cysticercosis when they swallow T. solium eggs, and kung may naiwan doon na egg sa kanilang digestive tract, it will develop into a larvae that lodge in the tissue, usually sa ating brain, eye, and muscle. So, kung nasa brain yung T. solium eggs, it is called neurocysticercosis. So, when larvae develops in the CNS, central nervous system, so symptoms are mimic epilepsy or a brain tumor. Laboratory diagnosis, with the use of computer tomography, CT scanning, or magnetic resonance imaging or the MRI, serological test, which is blood test para ma-detect yung presence ng antibodies or para malaman din natin kung exposed ba yung tao sa particular microorganism. So ito, example na picture, may tapeworm infestation sa brain. Sa mga mahilig naman kumain ng sashimi and sushi, this is the Diphyllobothrium latum, which is also known as fish tapeworm, found in trout, perch, and salmon. So, develop symptoms, you will have abdominal distension, belching, yung nag-burp, intermittent abdominal cramping, and diarrhea. Hydatid disease, so one of the most dangerous tapeworm is the Echinococcus granulosus. So the adult form lives in the intestinal tract of the carnivorous animals such as dogs and wolves. So maging infected yung tao from feces of a dog na kuwari nagkain siya ng dog, tapos yung dog naman nakakain ng flesh ng sheep or deer na may cyst form of the tapeworm. Itong sakit na to occurs most frequently sa mga tao lang nag-raise ng sheep or may farm, tapos nag-hunt siya or trap ng wild animals. But once naka-ingest ka ng eggs of Echinococcus, nang mag-migrate siya sa various tissues of the body, lalo na ang liver and lungs, kayo ng mga common sites. Then, kung may nakita na sila nga pwesto, pwede na silang mag-develop into a hydatid cyst. Diagnostic test, you have serology and x-ray exam. Treatment naman, dapat siya i-surgery and with the use of albendazole. Yan ang example picture, may cyst ng kanyang brain. Pinworms, pathogen Enterobius vermicularis. So this tiny worm migrates out of the anus of the human host to lay its eggs, causing local itching. Diagnostic test, so it usually based on finding eggs around the anus. Pwede ito ma-viewed by pressing transparent cellulose tape. So dapat naka-stick siya side down against the skin, transferring the tape to a microscope slide and viewing the slide under a microscope. Treatment is yung parental pamote. Hookworms. So hookworms infections were once a very common parasitic disease in the southeastern states. Pathogens Wernicketor americanus and Ancylostoma duodenale. The hookworm attaches to the intestinal wall and feeds on blood and tissue. Presence of large numbers can lead to anemia and lethargic behavior. Heavy infections can lead to pica. No mag-crave ka sa mga peculiar or unusual foods. Tapos, pica is a symptom of iron deficiency anemia. Diagnostic treatment is the microscopic exam. So, hookworm infections, diagnose sila by finding parasite eggs in feces and can be treated effectively with mebendazole. Ascariasis, pathogens Ascaris lumbricoides. So in the upper intestine, the eggs hatch into small worm-like larvae that pass into bloodstream and into the lungs. Symptoms, nausea and vomiting, diarrhea or bloody stools. Then in the lungs can cause pulmonary symptoms. Diagnostic treatments such as microscopic exams. Treatment, mebendazole. Whipworm. Whipworm infestations known as trichuriasis are widespread in tropical areas of the world, especially Asia. Pathogens, trichuria. So in southeastern states, children pick up infective eggs from contaminated soil. Pag nilagay nila sa kanilang mouth, so yung na-ingest nila yung egg, so it hatches and magpasok siya sa intestinal glands, then the worm positions itself and extends into the intestinal lumen and the hair-like anterior buried in the mucosa. So the worm lives there for several years as a tissue parasite. Habang nandoon siya, i-feed niya yung cell contents and blood. That is why trichuriasis can also lead to anemia and malnutrition, resulting in significant weight loss and retarded growth. Diagnostic treatment, microscopic exam of feces, treatment, albendazole and mebendazole. Trichinellosis, so pathogen is Trichinella spiralis, so formerly called trichinosis. So pagkain din ng undercooked pork is probably the most common mode of infection or other animals that feed on garbage. Sa pagkain din ng raw sausages or hamburger and even chewing fingernails after handling infected pork. Symptoms include fever, swelling around the eyes, and gastrointestinal upset. So small hemorrhages under the fingernails are often observed. Biopsy specimens as well as a number of serological tests can be used in diagnosis. Example is this ELISA. It is enzyme-linked immunosorbent assay that detects the parasite in meats has been developed. Treatment consists of administering albendazole or mebendazole to kill intestinal worms and corticosteroids to reduce inflammation. Amoebiasis or amoebic dysentery is a protozoal gastrointestinal infection that may be asymptomatic, mild, or severe. In some cases, it invades the colon wall, causing colitis, acute dysentery, or long-term chronic diarrhea. So, ang amoeba pwede siyang ma-spread via bloodstream, so the infection can also spread through the liver. In rare cases, it can spread to lungs, brain, or other organs. Patient care uses standard precautions for hospitalized patients. Gastrointestinal amoebiasis is treated with nitroimidazole drugs. These drugs kills amoebas in blood, in the wall of the intestine, and in liver. Geographic occurrence naman is amoebiasis occurs worldwide. Amoebiasis is caused by the parasite Entamoeba histolytica. Reservoir and mode of transmission. One is via ingestion of fecally contaminated food or water containing cysts. Second is sa mga langaw, nagatransport ng cysts from feces to food. 3. Via fecally soiled hands of infected food handlers. D. By oral anal sexual contact or by anal intercourse involving multiple sex partners. Laboratory diagnosis is microscopic observation in stained smears of fecal specimens. Importante ito siya sa mga mag-check para malaman nila i-differentiate yung E-histolica from other pathogenic and non-pathogenic intestinal amoebas. Then, pag may trophozoite sa red blood cells, meaning merong invasive amoebiasis. Balantidiasis, it is a protozoal gastrointestinal infection of the colon causing diarrhea or dysentery. So dysentery is cause bloody diarrhea, nausea, and vomiting. Patient care also use standard precautions. Tetracycline, metronidazole, and iodoquinol ang need. Geographic occurrence, although seen worldwide, balantidiasis is rare in the United States. Balantidiasis is caused by Balantidium coli. It is the largest of the protozoa that infect humans. Balantidiasis occurs more commonly in pigs. Mode of transmission includes pigs and anything that might be contaminated with pig feces. Example is drinking water. Transmission most often occurs via ingestion of B. coli cysts in fecally contaminated food or water. It is diagnosed by observing and identifying B. coli trophozoites or cysts in fecal specimens, which may also contain blood and mucus. Then we have cryptosporidiosis. It is a gastrointestinal infection caused by a coccidial protozoan. Coccidia are sporozoan, which means it is unicellular, parasitic, and capable of reproducing sexually and asexually. It may be asymptomatic or may cause diarrhea, cramping, and abdominal pain. Less common symptoms include fever, anorexia, nausea, and vomiting. The disease may be prolonged, fulminant, and fatal in immunosuppressed patients. Ito ay may mahina na immune system. Children who are 2 years of age or younger, animal handlers, travelers, homosexuals, and daycare center workers are particularly likely to be infected. Outbreaks in daycare centers are common, so also been associated with drinking water, recreational use water, and drinking unpasteurized apple cider contaminated with cattle feces. Patient care also is standard use precautions for hospitalized patients, add contact precautions for diapered or incontinent patients. Geographic occurrence, this disease has been reported worldwide. The largest waterborne outbreak that has ever occurred in the United States was the 1993 cryptosporidiosis outbreak in Milwaukee, affected more than 400,000 people. Cryptosporidiosis results from ingestion of oocysts of Cryptosporidium parvum. Oocyst is a cyst na merong zygote, and a zygote is a fertilized egg. Then, ito yung itsura niya. So, the parasite, meron siyang protected layer or shell para makasurvive siya outside the body for long periods of time. Reservoirs include infected humans, cattle, and other domestic animals. Meron din fecal-oral transmission from person to person, from animal to person, or via ingestion of contaminated water or food. Laboratory diagnosis, it can be diagnosed by microscopic observation, acid-fast oocysts in stained smears of fecal specimens. Sensitive and specific immunodiagnostic procedures are also available. It is a test to detect antibodies and parasite antigens. Then we have cycloporiasis. It is a coccidial gastrointestinal infection causing watery diarrhea. Mayari siyang 6 or more stools per day, nausea, anorexia, abdominal cramping, fatigue, and weight loss. May tendency din yung diarrhea lasts between 9 and 43 days in immunocompetent patients, yung may malakas ang kanilang immune system, and months naman in sa immunocompromised patients, which is yung mahina ang kanilang immune. Patient care also use standard precautions for hospitalized patients. Geographic occurrence, cyclosporiasis has been diagnosed in Asia, the Caribbean, Mexico, Peru, and the United States. The parasite is the ingestion of oocysts of Cyclospora cayetanensis, a coccidian. So, sa reservoir naman, include dito yung pag-inom ng fecally contaminated water, laboratory diagnosis made by microscopic observation, so it is 8 to 9 meter diameter, which are about the size of Cryptosporidium oasis. The oasis autofluorescence means may natural emission of light siya na bright green to intense blue under ultraviolet fluorescence when examined. Lastly is the giardiasis. It is the protozoal infection of the duodenum. Makita siya sa uppermost portion ng ating small intestine and may be asymptomatic, mild, or severe. Patients experience diarrhea, steatorrhea, which is loose, pale, or fatty stools, abdominal cramps, bloating, fatigue, and possibly weight loss. Patient care for giardiasis also use standard precautions for hospitalized patients, at contact precautions for diapered or incontinent patients. Geographic occurrence, this disease occurs worldwide. So ang tawag sa kanyang pathogen is the Giardia lamblia, also called the Giardia intestinalis, a flagellated protozoan attached by means of a ventral sucker to the mucosal lining of the duodenum. Mode of transmission, possibly makuha natin sa mga wild and domestic animals such as beavers have consumed water containing giardiasis and fecally contaminated drinking water and recreational water. Laboratory diagnosis, so it diagnosed by microscopic observation of stained smears of fecal specimens or duodenal aspirates. So gina-examine siya using the fluid from the duodenum para ma-check kung may signs of infections. The Giardia tropozoite has been described as resembling an owl face, a clown face, or an old man with glasses. So immunodiagnostic procedures are also available. So ito ang itsura ng giardiasis, parang teardrop shape siya, tapos sabi nila parang may itsura daw, that's why it contains 2 nuclei. And that's all. Thank you for listening.